Thank you for coming to my online talk. So when we think about a sort of simplified communication scenario, we often envision one sender sending a signal to one receiver. And for the purposes of my talk, I'm going to be talking about sexually selected signals with male signalers and female receivers. So here we have a male frog producing a call, which is then received by a female frog. But communication in the wild, it often doesn't take place in this very simple idealized scenario. It often takes place in communication networks in which multiple senders and receivers will be within earshot of one another at any one time. This can influence how communication plays out in interesting ways. So, for example, when receivers can receive multiple signals simultaneously or close in time, the temporal relationships amongst these perceived calls may influence how they're rated in terms of attractiveness relative to one another. And at the same time, they can now be comparatively evaluated, so a male's relative attractiveness may become more important than his absolute attractiveness. And at the same time, males will be within earshot of one another's signals too. And this can influence male signaling strategies. For example, neighbors may establish certain timing relationships with their signals, and they may increase attractive call properties in order to increase that relative attractiveness. And of course, as you add additional individuals to these communication networks, things get more and more complex. We were interested in how communication plays out within túngara frog choruses. So we had two broad questions. How do male calling strategies interact with one another and with the social environment? And how does this affect female preferences? So first, a little bit about túngara frogs. Males can produce simple whine-only calls, which are shown in subfigure A on the right here, and they can produce complex calls, which are whines typically followed by 1 to 3 chucks, as can be shown in B through D here. And females are 5-fold more attracted to complex calls than they are to simple calls. Males alternate their calls with rivals in smaller choruses, and males form dense choruses in the wild. So for example, in this image on the right here, you can see 9 calling males as highlighted by these blue stars, all in very close proximity to one another. And how male calling strategies play out in these larger aggregations hasn't been very fully studied. So to investigate how calling strategies play out in these larger choruses, we built some experimental choruses of 2 to 6 live males in this kind of hexagonal arrangement you see at the bottom of the screen. So in total, we had 69 males in 16 unique choruses. And for each chorus mate, we extracted the timestamps of all of their calls. So the first call we asked was, when does call overlap become unavoidable? So to check this out, we randomly selected a focal male in each chorus and then looked at the mean duration of the silent gaps available to him to call in. So that's shown on the y-axis, and then the mean time between silent gaps available to him to call in, which is on the x-axis. And then I've also put this dotted line here, which represents the average duration of a túngara frog call. So any of the data points below this suggest that the mean duration of silent gaps available to males in that chorus are not sufficient to insert a call without overlap. And so basically the takeaway here is that call overlap becomes unavoidable in any chorus larger than 3 males. So we saw that overlap occurs, and then we wanted to see, what does this call overlap look like? So to do this, we randomly chose 100 calls per male for each of the males calling within our choruses. And then we calculated how they related temporally to the call that directly preceded them by a chorus mate. And then we presented these in these ridgeline plots here for choruses of different sizes. And I've also superimposed a whine with a chuck of average duration to see whether that temporal relationship likely would have resulted in overlap. And so you can see in choruses of 2 and 3 males are pretty much waiting until the call that's occurring, the call that's preceding them by a chorus mate has finished, and then are placing their call after that call has ended. However, in these larger choruses, we see that males are calling simultaneously more often, as you can see by this increasing peak around about the 0 millisecond mark. And they're also increasingly inserting their calls just before the chucks of chorus mates' calls. So you can see it ends up looking something like this, as demonstrated by these blue calls on the left. The beginning of the following call's whine tends to overlap the leading call's chuck a lot of the time. And we also found that call overlap was more common among more distant chorus mates. So this quite striking difference in the temporal relationships amongst males in smaller and larger choruses sort of begs the question, why does call overlap look like this? So I actually think that these very different timing relationships actually result from males doing the same thing, which is waiting for quiet gaps in which they can call and then placing their calls there. And obviously in choruses of 2 and 3 males, these quiet gaps are true silence. However, silence gets rarer and rarer in these as choruses get larger and larger. Whereas at the same time, as I've highlighted here with this red circle, we know that whines actually get very low in amplitude right at the end, right before they give way to the chucks. So what I think is going on in these larger choruses is that actually, because true silence is so rare, this quiet bit at the end of chorus mates chucks becomes the most commonly encountered local amplitude minima, and therefore becomes an attractive place to place your call. And so this is why males call here in larger choruses. So we know that overlap of this stereotype nature occurs, so we wanted to see, does overlap influence female choice? And so we decided to build a temporal check preference function using phonotaxis experiments using the setup on the left, in which a female, shown as in a circle here, is placed equidistant between 2 speakers, each of them playing a whine chuck. And then the stimuli we used for this experiment are shown on the right here. So we can see that here, the leading call coming from one of the speakers leads the following call by 50 milliseconds. Here we see it leading the following call by 150 milliseconds, 150 milliseconds, and on in similar increments up to 750 milliseconds. For each of these treatments, we ran 35 females, and we analyzed female preferences for leading or following calls using two-tailed binomial tests. And the results of these tests are presented on the right here. So just a little bit about how to interpret this figure. On the y-axis is the probability of preferring a leading call. So any data points above this red chance line are preference for the leading call, whereas any data points below are preference for the following call. And then if you'll envision that this blue call here is the leading call in each treatment, each of these vertical gray lines represents when the following call began relative to that leading call in each treatment. So we can see that females strongly preferred the leading call when following calls began 50 milliseconds after the leading call and when they directly abutted the leading call. However, what's interesting is that when the following call began 250 milliseconds or 350 milliseconds after the leading call, females actually strongly preferred the following call. So this suggests that basically, as is shown on the left here, when any part of this first 150 milliseconds of the following male's whine overlaps the leading male's chuck, it seems that the females discriminate against the leading call in favor of the following call. And what's very interesting is that the type of overlap we saw in our larger experimental courses fit exactly this template. And so what might be going on here? So it could be that whines begin at their highest amplitude and then decrease. So maybe this first part of the whine is of sufficient amplitude to partially mask the leading male's chuck. Or it could be that important frequencies for mate recognition are contained in this first part of the whine. And so perhaps they take precedence during processing in the female perceptual systems when they occur simultaneously with the frequencies contained in chucks. And though these results were suggestive, we wanted to test this out in a more realistic setup. So for example, a female will seldom find herself just sitting equidistant between 2 males. In reality, she'll often be approaching a cluster of males from one side. And what's more, as we talked about, overlap was more common amongst more distant chorus mates. So oftentimes the following call is going to be perceived as much lower in amplitude by the female as is the leading call, or vice versa. We came up with this new setup which attempts to mimic more closely what a female might encounter when approaching a chorus in the wild. So in this, we have the female on the right side of the chamber and the 2 choice speakers on the left side of the chamber, each playing a whine chuck. So these are shown by the black squares. But we also have an interference speaker, which is placed on the ceiling above these choice speakers, projecting sound directly down. And this is also playing a whine chuck. And as you can see from the stimulus on the right here, the interference speaker is playing its whine chuck as overlapping one of the choice speakers in the way that we saw in our experimental choruses. And we were also able to manipulate the amplitude of our interference speaker so that it would be perceived by females as coming from 1 meter beyond the choice speakers, or 2 meters, 3 meters beyond the choice speakers. And what we found was that the attractiveness of overlapped calls was significantly reduced even when overlap came from up to 3 meters away. So you can see in the figure on the right here on the y-axis we have the probability that females preferred the non-overlapped call, and in all distance treatments the results were way above this chance 0.5 line. And so our results so far suggest that chucks are at risk in any chorus larger than 3 males within 3 meters, which as we've seen is not a tall order for the túngara frog. And so these results cause us to wonder, do additional chucks safeguard calls against complete chuck overlap in these larger choruses? So as we saw, it seems to be this first 150 milliseconds of following calls that has the capacity to reduce the attractiveness of leading calls when it overlaps the chuck. And we also know that males can append additional chucks to their calls, as shown in this figure, with each additional chuck lengthening the chuck containing portion of the call, which could reduce the likelihood that every single chuck is overlapped by that 150 milliseconds of chorus mates whines. And we also know that males increase call complexity as chorus size increases. So to investigate this, for each frog in our experimental choruses, we randomly chose 10 of his whine with 1 chuck, whine with 2 chuck, and whine with 3 chuck calls. And then we used the timestamp of individual call elements to determine whether each chuck in those calls was overlapped by that first 150 milliseconds of a chorus mate's whine. And then we built a logistic regression, with the response variable being whether each call had at least 1 chuck free from such overlap, and then our predictor variables being chorus size, call complexity, and their interaction. What we found was that more chucks per call increased the probability that at least 1 chuck remained unmasked by that first 150 milliseconds of chorus mates whines, especially so in larger choruses. So you can see in this figure on the right, on the y-axis, we have the predicted probability that a call had at least 1 chuck free of overlap. We have chorus size on the x-axis. And you can see that for 3 chuck calls in kind of orange at the top, there's not much of a reduction in terms of the probability that a call had at least 1 chuck free in any chorus size. Whereas in 2 chuck calls in green, there is more of a drop as chorus size increases. And in 1 chuck calls in blue, there's a much more precipitous drop in the chances that a call will have at least 1 chuck free from overlap in these larger choruses. But we also wanted to verify experimentally that this influences female preferences. So we did phonotexis test using the setup on the left again, but the stimuli used in these sets of tests were 1 choice speaker was playing a whine with 3 chucks and 1 was playing a whine with 1 chuck, and both calls were overlapped by the interference speaker in that way that we've seen. And again, we varied amplitude of the interference speaker to seem 1 to 3 meters beyond the choice speakers, and also included a control treatment in which neither call was overlapped by the interference speaker. And so females preferred whines with 3 chucks over whines with 1 chuck when both calls were overlapped, even when that overlap came from up to 3 meters away. However, when neither call was overlapped, females showed no preference for calls with more chucks. So how does this fit with previous results? So calls with more chucks have been shown to be more attractive in certain experiments, though many studies have found no benefits of additional chucks. I think what our results suggest is that a key benefit of additional chucks only becomes apparent when looking at túngara frog communication in a chorus setting, in which these additional chucks will safeguard calls against that detrimental overlap that seems to be ubiquitous in these larger choruses. And I'd like to thank my lab, various funding bodies, and thank you all for watching.